Hey everyone, we are going to continue with fatty acid catabolism. Today we'll be focusing on the actual steps that occur in the mitochondria to break down fats. Remember that last session we focused on the actual complex protein that is required to do each of these steps. It's a protein that has three main subunits, if you'll remember, each responsible for a portion of this actual process. So here is an example of a fat that has entered into fatty acid catabolism or beta oxidation. That is the name of the series of events that take place to oxidize or break down a fat. Here we're looking at palmitoyl-CoA. It is a 16-membered lipid. I'm going to highlight that. And you can see we're looking at the end of it. So notice here is that thiol-CoA bond that has been added onto the fatty acid to make it a acyl-CoA group. And what we're looking at is the carbon directly adjacent to the carbonyl. This is referred to as the alpha carbon when talking about beta oxidation. And one more carbon over is referred to as the beta carbon. Now, it is important when talking about how long these carbons are to remember, or these lipids are, that this carbonyl carbon is also counted. It is still a carbon that is part of the whole chain of the lipid. You can see the first step is a dehydrogenase step, which we are familiar with after talking about the citric acid cycle. This is going to remove hydrogens. This makes a double bond, and it is also mainly removing electrons. They are going to be removed to the electron carrier, FADH2. So again, this is an acyl-CoA dehydrogenase step, and you can see what has happened is we've removed electrons here from this portion between the alpha and beta carbon. Those have then reduced an FAD to an FADH2, which can, of course, go off to the electron transport chain eventually to make energy. The second step is a hydratase step. It's actually the addition of water. You can see it's being added to the double bond that we just formed. So here is our double bond that was made between that alpha and beta carbon, and we are hydrating it. So it's going to add a hydrogen on one side and an OH on the other side. Note that the OH group is actually forming on what was the beta carbon. So at this point, you can see we've essentially regenerated that CH2 carbonyl group that was there at the very beginning and lost some electrons, and through this process also made an OH group on our beta carbon. This third part here is going to be an additional dehydrogenase step. So there are two dehydrogenase steps. The first one, the acyl-CoA dehydrogenase. Again, this is saying that it's doing a dehydrogenase step on the original acyl-CoA. And our second dehydrogenase step is going to occur now on that OH group that we just placed. So it's going to be on the beta hydroxy acyl-CoA. Thus the name of this enzyme, beta hydroxy-CoA dehydrogenase, meaning the hydroxy group on the beta carbon of our acyl-CoA. We're going to remove hydrogens from that position and electrons. In this step, though, we are removing those electrons to an NAD to reduce it to an NADH. So again, the first step, we produce an FADH2, and the second step, we produce an NADH. Now, after we have further oxidized our lipid, you can see that OH group became a carbonyl. The last portion is called acyl-CoA acetyltransferase. It's also referred to as thiolase. Notice the ace portion in here, thiolase, refers to cleavage of the molecule. At this point, we have situated our fatty acid to have a whole acyl-CoA group. Notice 1, 2 carbons and our thiol-CoA. This is the backbone of an acetyl-CoA. And we've made it so that when we cleave the lipid, we now are starting with an identical structure to what we started with in the beginning. See that carbonyl right there. And this chain that's left over can continue now going through the process of breaking off chunks of 2 carbons. So that's exactly what's going to happen. We're going to break off this acyl-CoA, and in doing this, notice we do need to take a new thiol-CoA and add it on to the end of our lipid that was left over after breaking off one acyl-CoA. So here's our new thiol-CoA bond on our carbonyl carbon that used to be our beta carbon. And now this is a C14 lipid that can continue through beta oxidation. Essentially, this will go all the way back up to the beginning and go through the process again, this time breaking off another acetyl-CoA. So let's talk about each of those steps, the enzymes involved, and a little bit about more detail of what's going on. So again, step 1, remember, in this beta oxidation process is the dehydrogenation of an alkane to an alkene. We're forming a double bond that is on the beta carbon, catalyzed again by acyl-CoA dehydrogenase, and this is on the inner mitochondria membrane. This particular enzyme can work on different types of lipids, and it has different names for each size. So there is a very long chain, acyl-CoA dehydrogenase, or a abbreviated as AD. So very long chain AD, medium chain AD, and short chain AD. And you can see the variety of carbon lengths that those can catalyze. These result in a trans bond. So again, when this bond is actually made during beta oxidation, I want to be clear that it is a trans double bond, which is different from when we have naturally occurring unsaturated fatty acids. So far, we've just talked about what happens when you have a fatty acid that has all single bonds. And we'll get to what happens when you have an unsaturated fatty acid, which is a fatty acid that already has a double bond. But I want to remind you that those fatty acids, naturally occurring fats with double bonds in them already, have cis double bonds. So this process results in a double bond that is unlike the natural occurring double bond. This particular enzyme is also analogous to another enzyme we have seen, which is succinate dehydrogenase reaction, and that occurs in the citric acid cycle. You can see it is using FAD in this particular enzyme, succinate dehydrogenase, but that FAD is actually bound to the enzyme. It is not free floating. And those electrons can be directly tunneled to the electron transport chain from succinate dehydrogenase. That is one of the special things about succinate dehydrogenase, is that it is also part of the electron transport chain. But its interworkings are very similar to acyl-CoA dehydrogenase, but remember that acyl-CoA dehydrogenase does utilize an FAD that will be free-formed and that can then go to the electron transport chain. Here's a close-up of that FAD cofactor. I wanted to give it a little bit of time since we have been talking about NADH a lot, and looking now at beta oxidation and citric acid cycle, we are getting more familiar with our second most used electron carrying energy molecule, which is FAD. So FAD is based in a vitamin. This is riboflavin vitamin B2, sometimes abbreviated as FMN. Its whole name derives from the word flavin adenine dinucleotide and flavin mononucleotide. So when it is the full FAD, it is the flavin adenine dinucleotide. So we can see here is the actual riboflavin vitamin portion of it, the flavin mononucleotide here, with our phosphate. And when it has an adenine group, and you can see here is a ribose attached to it. So this is a full nucleotide bottom. This is the FAD portion. So it is based in a nucleotide structure, just like our NADH, but its top portion, this riboflavin portion at the very top, is what is doing all of the work. You can see this semiquinone type structure here is where our electrons are actually being carried. It can grab onto or remove off 2 electrons in total. Its fully reduced form being FADH2. Okay, so step 2 in the process, remember, is this hydration. hydration of the double bond, that alkene we just formed on the beta carbon. This is catalyzed by enyl-CoA hydrodase, which also has two forms. There's a soluble form. This is a form that is not bound up to that complex protein for beta oxidation. So this is a form that is just solubly floating in the mitochondrial area. It is also sometimes referred as chronotase. And then there is the membrane-bound one, the one that is part of this complex that is utilized for long chain. I want to remind you that the trifunctional complex that we've talked about, the membrane-bound version, only works on these longer chains of lipids. And if you do have shorter chain lipids, as noted in last class, they are worked on or broken down using those soluble versions of the same enzymes. So during the hydration portion, again, water is added on the beta carbon, yielding an alcohol. And this reaction is analogous, again, to another reaction we've seen in citric acid cycle, which is the fumarase reaction. It also results in the same stereospecificity. When you break open a double bond and you add now a new element, so here we're adding on an OH group, this is actually forming a chiral center briefly, and it is in the L form. So again, we've talked about our formation of double bond, which is a trans double bond using acyl-CoA dehydrogenase, and the formation of this OH group on the beta carbon, which is in the L formation, just like in fumarase. Step 3 is our second dehydrogenation step. This time it's the dehydrogenation of an alcohol instead of a single bond to a double bond, catalyzed by beta hydroxy acyl-CoA dehydrogenase. This again uses that NAD as the acceptor instead of FAD. And this enzyme, beta hydroxy-CoA, only recognizes the L form of that isomer. So I just said our previous enzyme will only form the L form of that alcohol. And thus, to make sure that we are only working on the proper fatty acids, this will only accept and fit the L version of that alcohol. This is analogous to the step after fumarase, which is malate dehydrogenase in citric acid cycle. Remember again that fumarate and malate reactions do a very similar thing. They form an alcohol, which is then re-dehydrogenated into a carbonyl. And our last step is the step in which we are going to cleave an acetyl-CoA that can be used for energy and finalize and prep the rest of the lipid for potential further beta oxidation. So this acetyl-CoA acetyltransferase is one of the more important steps. Thiolase is going to take the carbonyl carbon on the beta position of that fatty acid, which will become the new kind of end group of the leftover fatty acid. And during this process, we're going to cleave off a whole acetyl-CoA. So this, as I noted, can happen numerous times. You're going to have what's called passes of the same fatty acid through beta oxidation as it sequentially cleaves off an acetyl-CoA until essentially there's nothing left of it. If we, for example, started with palmitic acid, which is what I showed before, this is a C16, has 16 carbons. You would basically repeat those 4 steps 6 more times, so 7 in total, resulting in 8 molecules of acetyl-CoA. So remember that the last step is going to, it'll be 4 carbons on that last step, and it will then cleave into an acetyl-CoA, and we'll only have 2 carbons left, which will be another acetyl-CoA. So a 16 carbon fatty acid requires 7 cleaves, resulting in 8 molecules of acetyl-CoA. Here's the key part: 7 FADHs and 7 NADHs, because remember, we only form those during the process. So if we only have 7 cleavage processes, then we're only going to get 7 of those energy molecules. All of that acetyl-CoA is where we are getting the energy. And all of that acetyl-CoA is going to go into the citric acid cycle, where it, of course, can make more GTP, more NADH, and more FADH. So from fatty acids, we've generated a lot of essentially NADH and FADH through the breakdown of all of these acetyl-CoAs. And those are going to eventually funnel into the electron transport chain, where we will see how they dump off those electrons to make more ATP. So I wanted to show you a visual of the parallels I was talking about while talking about beta oxidation. I noted a lot of steps that were very similar between beta oxidation and the citric acid cycle. So here I'm showing you beta oxidation on the left. These are the 4 steps of beta oxidation. And I'm also showing you 4 steps of the citric acid cycle. This is from succinate to oxaloacetate. So it's like the second half of the citric acid cycle where we're regenerating oxaloacetate. And you can see that there are portions of it that are pretty much identical. We have a step in which we have a single bond that is made into a double bond through the generation of FADH. This is then hydrated with a water to make an alcohol. That alcohol is then reformed into a carbonyl while generating an NADH. There is another process that we will look at later that has this exact parallel, and it is in the oxidation of some particular amino acid. So oxidation of isoleucine or valine during a process of branched chain amino acids. So again, just a reminder that we'll be seeing that again, that reaction sequence pop up. So we talked about how saturated fatty acids are broken down very easily. They're going to go through that 4-step process of beta oxidation until we have cleaved them into little packets of acetyl-CoA, which can go into citric acid cycle. But what happens when you suddenly have a double bond in your chain? Remember that naturally occurring unsaturated fatty acids contain cis double bonds, which would not work for our enol-CoA hydratase, which does take a double bond and make it into that alcohol. So to kind of prep the lipid to then go into the rest of the steps of beta oxidation, we need some additional enzymes. And these are going to help turn our cis double bond into a trans double bond. This uses an isomerase that takes it from the cis to the trans, and a reductase, which reduces the cis double bond to the correct position. Monounsaturated fatty acids, you'll see these are fatty acids that only have one double bond in them. They only require the isomerase, while polyunsaturated fatty acids are going to require both enzymes, the isomerase and the reductase. So again, just a reminder, here is the main layout of what you would do for a saturated fat. And again, if we have a double bond right here at that beta position on the beta carbon and over, and it is in the trans formation, then you can just start at the enol-CoA hydratase step and continue through beta oxidation. So here's an example where we have a monounsaturated fatty acid, and you can see it's going along cleaving off portions. Here's our first set of 2 carbons. You can see here's 2 carbons, and we've done a first cleave. That's 1 acetyl-CoA. Another 2 carbons, another cleave. Another 2 carbons, another cleave. At this point, you can see we now have a carbonyl here. This is our alpha carbon. And starting where our double bond is our beta carbon. This is the exact scenario you want to have your double bond sitting in. So when it starts at the beta carbon like this, then we have it in the correct position. The only problem here is that it is a cis double bond and not a trans double bond, which we need to put into our beta oxidation. So this is going to utilize an enol-CoA isomerase instead of doing our initial acyl-CoA dehydrogenase step, which we would if we had a saturated fatty acid. At this point in the chain, we will use our enol-CoA isomerase. And what it will do is just make our double bond from a cis to a trans. Well, at that point, we have the perfect situation. We have a trans double bond starting at the beta carbon, and we can continue with the enol-CoA hydratase step and continue going through to cleave off that next acyl-CoA. So beta oxidation can just continue going through. What I want you to note is that once this process cleaved up into the point where it had to deal with the double bond, note that we did have to skip this acyl-CoA step, right? We don't have to make a double bond because we already have a double bond. We just have to rearrange it to get it into the enol-CoA hydratase. But by skipping that step for that one point, we do not generate an FADH2. So essentially, when you have a monounsaturated fat, you're going to do your cycles normally, except for one of those cycles in which you are dealing with that double bond. And during that cycle, that cleavage cycle, you skip the step where you would generate FADH2. So you result with one fewer FADH2 with a monounsaturated fat. ultimately you see that these unsaturated fatty acids are actually going to be less energetically favorable and that they're giving us less ATP in the long run. So, to give you an example of what those numbers would look like, if you had a fatty acid that was a C18, it was 18 carbons long, it would generate 9 acetyl CoAs. Remember, this is where each acetyl CoA is 2 carbons long, so it's just half of that. Now, normally, if it was saturated, it would take 8 cleavages to break this C18. That would form 8 FADH2s and 8 NADHs. If that C18 fatty acid was monounsaturated, it had one double bond, instead it would form 7 FADH2s and 8 NADHs. So the only difference here is that we have removed one FADH to deal with that step where we are skipping the process of dehydrogenasing to make a double bond. Now let's look at a polyunsaturated fatty acid. This gets a little bit more complicated because we are going to need an additional enzyme to deal with 2 double bonds next to each other. Here we're looking at linolyl CoA, and it has a double bond starting at the 9 position and another double bond starting at the 12 position. With linolyl CoA, we have now cleaved off a couple of our acetyl CoAs. So 3 acetyl CoAs have been cleaved off. We've gone through 3 cycles of beta oxidation. And at this point, we now have a 12 carbon long fatty acid. And we've essentially gotten to the point where our double bonds start. So again, here is our alpha carbon and here is our beta carbon. You can see the beta carbon is where our double bond starts. So this is good, but it is a cis double bond. So just like last time, we're going to use the isomerase. That makes the double bond go from a cis double bond to a trans double bond. But during this process, it's actually, we're going to shift it over right here between that alpha and beta. And then we can re-enter it into the cycle just like last time, cleave off another acetyl CoA. So again, after the isomerase, we can continue breaking off this acetyl CoA. So during this step, just like last time, we will miss out on one FADH2, but we'll start to continue on to the next step. So now we have cleaved this whole portion off, cleaved it here essentially where that double bond was. And that left us with a carbon here, 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 and here. This is our new setup, which you see below. And as that fat enters, it does not have a double bond that is close enough to just isomerase. So it will go through the normal phase of dehydrogenating the alkene to a double bond. And that's actually already shown. So again, here is our new alpha carbon, our new beta carbon, and they have formed a double bond between them that is in the trans formation. And this is, again, just going to happen normally because of that portion that was broken off only has a single bond between it. We haven't kind of gotten to that next double bond. Now, to deal with this positioning where we have this double bond on our alpha and beta, but right next to it, we now have another cis double bond right here. So this situation we did not see before. In fact, if you look upward when we did our first isomerase, it separated out our trans bond fairly away from that double bond. So to deal with this positioning, we are now going to utilize the next enzyme that is required for a polyunsaturated fatty acid. This is this dienyl-CoA reductase. Notice the dienyl double enol. So we have a double bond situation. This generates a NADP. So it's going to actually use up an NADPH to make an NADP. Remember that when we were talking about the pentose phosphate pathway, the PPP pathway, we noted the generation of NADPH can be used during lipid synthesis and breakdown. So here is one of the cases in which NADPH is being utilized. And it's to rearrange these double bonds so that we don't have this situation where we have a trans double bond and this cis double bond back to back. This has been rearranged into one double bond that is now situated in the correct position. Just like it was up here. And it can be moved to finalize our oxidation with another enyl-CoA isomerase. So again, as soon as you start off with that second double bond, we're going to have to do the reductase, which uses an NADPH, and then follow it by an isomerase. We now have it all situated to go into the rest of beta oxidation for the rest of the cycles and generate the acetyl-CoA. Okay, so again, first double bond that we encounter in a polyunsaturated requires isomerization, and that is going to limit our FADH, right? Second requires reduction plus isomerization, which instead just uses an NADPH. So I'm going to give you kind of a total overview of what that looks like when you do have a polyunsaturated fat. So again, I'm going to give you the example of a C18 fatty acid, where you would generate normally 9 acetyl-CoAs, 8 FADHs, and 8 NADHs. If you had a polyunsaturated C18, so now it has 2 double bonds in it. Instead, again, we lose out on 1 FADH for that first double bond. You still make the number of NADHs. And for the second double bond, we also are going to use up an NADPH, so essentially generating an NADP plus. Now, most of our fatty acids are going to be even numbered. I've kind of skipped over this fact that I've been giving you fatty acid chain numbers that are all even numbers, which is great because acetyl-CoA is made of 2 carbons, and we can evenly divide that fat into 2 carbon chunks. So what happens when you have an odd numbered fatty acid? If you can't cleave it into an even number of 2 carbon chunks, we have to kind of work around this. So there are some cases where we could digest odd numbered fatty acids. Um, we normally deal with even number fatty acids, but some plants and other organisms do synthesize odd numbered fatty acids. What is going to occur is a 3 carbon compound, propionyl CoA, will be generated at the very end of our beta oxidation. So instead of finalizing in one last acetyl CoA, it will finalize in a propionyl CoA. As a note, lots of ruminant animals, this is like cows, for example, use bacterial metabolism to produce propionyl CoA. So because cows are eating plants, plants make some odd chain fatty acids. Those cows need to then break down those odd chain fatty acids, and they use bacteria in their stomach to help break it down. But in doing this, they produce a lot of propionyl CoA. So you would actually expect cow metabolism to have high propionyl CoA, while as human, for example, should not have high levels of propionyl CoA. So what happens when you do generate a propionyl CoA? At the end of your beta oxidation, how can it then be used for further metabolism? Well, propionyl CoA can be carboxylated. So propionyl carboxylase is actually going to utilize carbon dioxide via bicarbonate. It's going to use some ATP and a biotin. Remember, biotin is helpful in carrying CO2. And we're going to add on an additional carbon. So now it's a 4 carbon chain, this methylmalonyl CoA, which can then be rearranged through methylmalonyl CoA epimerase into eventually succinyl CoA. Succinyl CoA, remember, is part of the citric acid cycle. I want to note that this whole process is reliant, especially this rearrangement, on coenzyme B12, because that is where our CoA is coming from. This whole CoA portion that is then added on, which occurs during this methylmalonyl CoA mutase. This is the rearrangement, but the actual epimerase is going to take that carbonyl carbon, rearrange it with our CoA here. So again, this is just a close-up. We're going to take our propionyl CoA, add on a CO2 using biotin, and we're going to use up energy, use up an ATP. Then there's kind of like a 2-step rearrangement, the epimerase followed by the mutase, which is moving this CO2 that we just made and the thiol onto that CO2 around. And then a further movement where we're going to swap this hydrogen and that whole thiol carbonyl group around. So just a lot of rearrangement to then make succinyl CoA. Again, as noted, this isomerization on the propionate requires coenzyme B12. It is very important to utilize this coenzyme to help with the actual swapping of that whole carbonyl thiolate group and the hydrogen. Here's what coenzyme B12 looks like. It's a very complex coenzyme. It's very large, and it has a cobalt center. It looks very similar to the heme molecule. It does have a corrin ring system, which is essentially the same backbone of what you see with heme. It has this deoxyadenosine on top, so it has a nucleotide that is actually kind of coordinating with the cobalt in the center. And on the bottom, we have a dimethyl benzimidazole, ribonucleotide. So this is another similar nucleotide backbone. It doesn't have an actual adenine or an adenosine as we see on the top portion. And this one is interesting because it is both on one part covalently linked to the ring, but then this nitrogen is again going to be coordinating with the cobalt in the center. This reaction is very interesting in how it works to facilitate the group exchange. So you can see here is our whole complex. I'm just showing you the cobalt center. And in particular, this dimethylbenzimidazole, ribonucleotide portion. So here we see our carbonyl or our cobalt center that is going to be normally bound to this deoxyadenosine. Here it is normally. It's going to actually be cleaved into a free radical portion and then that whole cobalt portion. The substrate that we are interested in kind of swapping the two portion enters. And what happens is it's going to react with that free radical. So the hydrogen is going to help get rid of that free radical. And now our substrate becomes a free radical. There is then going to be a rearrangement of the radical in which we have an electron radical that is now swapped over to where our X, whatever X was, in this case, it's that carbonyl thiol group. And then there's going to again be a movement of a rearrangement in which we have now hydrogen that is replacing onto our product off of the B12 complex, forming our final swapped product. And now at this point, we still have a radical deoxyadenosine and the cobalt backbone. And this is then reformed, this bond between the deoxyadenosine and the cobalt is reformed to stabilize it. So ultimately, the cobalt is actually acting as a means to facilitate radical chemistry to then swap that hydrogen and our carbonyl thiol group. It's a very interesting chemical mechanism that occurs in this process. Okay, lastly, I want to focus on a type of group that is important in the formation of beta oxidation and utilized in other areas of metabolism, and this is ketone bodies. You may be familiar with them if you know anyone who is doing ketosis. So Acetyl-CoA, when it enters into the citric cycle, requires oxaloacetate, right? We're going to take acetyl-CoA, condense it with oxaloacetate, and then it can be further broken down to make more NADH, FADH, and GTP. But what happens when you make a lot of acetyl-CoA? So you are breaking down fats, breaking down lipids, but you don't have oxaloacetate if it's depleted. For example, if it's depleted to make glucose. Well, to break down, to help continue breaking down all of that built up acetyl-CoA, we need an oxaloacetate so that all of the acetyl-CoA that's building up from lipid breakdown can go into the citric acid cycle. So that acetyl-CoA, instead of going into the citric acid cycle, can be converted into something called ketone bodies. The reason this might occur again is if you're doing a lot of lipid breakdown, you have a buildup of acetyl-CoA, but you also are using your oxaloacetate for gluconeogenesis or for some other reason. This is helpful because it is going to free up all of the coenzyme A, and that coenzyme A can continue being used for beta oxidation. So we can kind of keep breaking down fats if we need to. And for the interim, we can kind of store that acetyl-CoA in these ketone bodies. These ketone bodies don't use coenzyme A. They kind of still have the energy that is there, and then they can, if needed, be turned back into acetyl-CoA later to be broken down through the citric acid cycle, or they can be used in other processes. So the three forms of these ketone bodies that you'll see is acetone, acetoacetate, and beta hydroxybutyrate. These can, they're formed in the liver, they can leave the liver. So to generate a free CoA and these then ketone bodies from acetyl-CoA, we're going to take two acetyl-CoAs and use thiolase. Thiolase is the same enzyme that is used in beta oxidation. It's that last enzyme that cleaves off the coenzyme A from our final, that final position on the lipid, and it kind of cleaves that lipid piece. So we're going to take two acetyl-CoAs, cleave off one of the coenzyme A's, and these are then going to be joined together. This is called acetoacetyl-CoA. So acetoacetyl-CoA is then going to react with one more acetyl-CoA and some water and an enzyme called HMG-CoA synthase. This is going to, as you can see, essentially generate on another set of carbons. So another two carbons from our acetyl-CoA is added on. That CoA on the acetyl-CoA that was added is released. And we now have this 6-carbon molecule called beta hydroxy beta methyl glutarol CoA, HMG-CoA. HMG-CoA is also an intermediate in the biosynthesis of sterols. So when we start talking about things like cholesterol, and steroid molecules, this HMG-CoA is an important molecule in that processes. HMG-CoA can then actually be lysed. It is kind of almost in reverse, can be broken back down into an acetoacetate. So a little bit different here because we did have an acetoacetyl-CoA. We go through this intermediate of HMG-CoA, which will then break off a whole acetyl-CoA, leaving this 4-carbon molecule, but notice it does not have a coenzyme A on it. And this is acetoacetate, excuse me. The reason we have to break it to this point where there is no coenzyme A on it is because coenzyme A is going to limit molecules from actually leaving. So to be trafficked outward into other tissues, the coenzyme A has to be removed. So once you form this acetoacetate, this is one of those ketone bodies. It can then be formed into one of the two other ketone bodies through other enzyme processes, either through losing a CO2 to form acetone or through the use of an NADH to form beta hydroxybutyrate. Beta hydroxybutyrate can actually travel through the blood. And just to note, any of that CO2 that is removed to make acetone also has to then get released in the blood. And acetone itself is actually removed as a gas, so it can be exhaled. The acetoacetate and beta hydroxybutyrate can also be trafficked to the brain, where they can be used for energy production in the brain. So here's kind of a close-up of that. The liver is our source of ketone bodies. This is where they're going to be generated. Again, it's going to occur when we have a buildup of acetyl-CoA from fatty acids, but that acetyl-CoA does not have enough oxaloacetate to go into the citric acid cycle. Again, that's usually happening when oxaloacetate is being used for gluconeogenesis to make glucose. So that's why you see this occur in fasting situations. If you have a situation where you're low on sugar, you're not ingesting a lot of sugar, but you are ingesting or using a lot of fats, you would see this exact scenario occur. You would have a buildup of acetyl-CoA from breakdown of fats, and you would have a lowering of your blood glucose. So you have to use oxaloacetate to make glucose through gluconeogenesis. This is also important. We're going to have to make glucose, remember, for things like brain and other tissues because they rely solely on glucose. That acetyl-CoA can then in the liver be made into ketone bodies. And one of the reasons we want to make them ketone bodies instead of just leaving them as acetyl-CoA is so that that coenzyme A piece can be kind of removed off, so it can be continued to be utilized in the further breakdown of more fatty acids and beta oxidation. All of these ketone bodies, again, can be utilized for energy in specific tissues like the heart, muscle, kidney, and brain. How are they used as fuel? Well, once we generate ketone bodies in the liver, again, they can just be removed via breathing them out, or they can go to tissues to be used for fuel. So for example, beta hydroxybutyrate or acetoacetate can then be transferred back into other cells after they have gone through the blood, where they will then go through the reverse, essentially. They'll have CoAs added back onto them. They'll go through beta ketoacetyl-CoA transferase and the thiolase to then break them back into acetyl-CoAs, which can be used for energy in those tissues. So again, just to kind of summarize, why do we use ketone bodies? Essentially, they're kind of used to temporarily shunt off excess acetyl-CoA so we can keep making ATP. They can easily move through the bloodstream, which is why you'd want to temporarily put them into this form. but ultimately they're going to go back into tissues where they can then be turned back into acetyl CoA once the coenzyme A and once we have citric acid cycle ready to then make more energy through breakdown of acetyl CoA. One very important thing about ketone bodies is they can pass through the blood-brain barrier. So they are often talked about when utilized in brain chemistry because they can kind of make it through the blood-brain barrier into the cells, neurons, et cetera, glial cells, to be used for energy breakdown in that system. And that's it for chapter 17. Again, things we summarized here are a talk on general in fats, how they're an important energy source. We talked about the process of beta oxidation, the 4 steps to make acetyl CoA from fatty acids, how lots of NADH and FADH form from this, and how they can then yield ATP as noted from the electron transport chain. How acetyl CoA formed in the liver can then either be oxidized by the citric acid cycle or in cases of fasting and starvation be converted to ketone bodies to serve for other fuels and tissues.